I mean, just stop and think about that for a second. You get a paper cut, and a few days later, it's just gone. Your skin fixes itself. You're constantly replacing old cells. So how in the world does your body manage to make billions of perfect new copies on such a massive scale, every single second of every day? Well, the answer is mitosis. And no, it's not some dry, boring term from a textbook. It's actually this elegant and incredibly precise dance, a sequence of steps that make sure every single new cell gets an exact copy of your genetic blueprint. Okay, to really get a grip on this, we're going to use a simple analogy. Just imagine that a cell's nucleus is a giant library, and all of its DNA, your genetic code, is all the books inside that library. So if you wanted to build a second identical library, what would you do? First, you'd have to copy every single book, right? That's what the cell does when it replicates its DNA. Then you'd pack all those books into dense, easy-to-move stacks. The cell does that too. We call those stacks chromosomes. Then you'd have to move those stacks with perfect precision and finally build two new identical libraries. That's exactly what a cell is doing during mitosis. So here's the formal definition. Mitosis is the division of the nucleus, our library's main office, to create two new genetically identical nuclei. You can really just think of it as the ultimate cellular copy machine. This whole cellular dance can be broken down into four main acts. So let's walk through them one by one, and we'll keep using our library analogy to see exactly what's going on at each stage. The four stages are prophase, metaphase, anaphase, and telophase. And a really easy way to remember them is just prepare, align, separate, and rebuild. Let's see how this all unfolds. We kick things off with prophase, the prepare stage. In our library, this is when all the books are taken off the shelves and packed into those dense stacks. In the cell, the long, stringy DNA starts to coil up super tightly, condensing into these visible structures we call chromosomes. At the same time, the walls of the library, the nuclear envelope, start to break down, and the cell begins to assemble the moving machinery called spindle fibres. Now, here's a key detail. Each of these chromosomes is actually made of two identical copies, called sister chromatids. Just picture the original book and its perfect photocopy held together by a single clip. That clip is called the centromere. And all this prep work leads us to the next stage, which is, incredibly orderly. This is metaphase. The spindle fibers get to work and move all those chromosomes so they line up perfectly, single file, right down the middle of the cell. It's like taking all your packed up stacks of books and arranging them neatly on a long table, making absolutely sure none are missed before you start sorting them. And then, bam, we hit anaphase. This is the most dramatic step. The spindle fibers act like little ropes that suddenly start shortening, pulling with force. This yanks the sister chromatids apart, splitting them and dragging them to opposite ends of the cell. It's the moment of separation. One complete set of books goes to one side of the room, and the other identical set gets pulled to the other side. Remember that clip I mentioned, the centromere? This is when it splits. And that is the absolute critical event here. This is what guarantees that each new cell is going to get a complete and perfect set of genetic instructions. Finally, we get to telophase, the rebuilding stage. At each end of the cell, a brand new nuclear envelope, new library walls, forms around the complete set of chromosomes. And the chromosomes themselves can finally relax. They uncoil, going from those tightly packed stacks back to being readable books on the shelves. So now we have one cell, but with two identical nuclei inside. But hold on, we've got two perfect libraries, but they're still inside one giant room. So how do we get two completely separate cells? Well, that needs one final step that actually happens right alongside telophase. That final step is called cytokinesis. This is the physical division of everything else in the cell, the cytoplasm. In our analogy, this is like literally building a wall right down the middle to create two distinct, separate, fully functional library rooms. Or, in this case, two new daughter cells. What's pretty cool is that this final split happens a little differently depending on the type of cell. Animal cells kind of pinch inward from the outside, almost like pulling the drawstring on a bag until it closes in the middle. But plant cells, they have those rigid cell walls, so they have to build a new wall, called a cell plate, right down the middle, starting from the inside and growing out. Now, with billions of these divisions happening all the time, you have to wonder, what's the potential for error? It's huge. A single mistake in this copying process could be disastrous and can lead to diseases like cancer. So how does the cell keep that from happening? The answer is a series of cellular checkpoints. You can think of these as incredibly strict quality control inspectors. Before the cell moves to the next stage, it gets hammered with questions. Is the DNA fully and correctly copied? Are all the chromosomes attached to the moving machinery exactly right? Is there any damage to the DNA? If the answer to any of those questions is no, the whole process just stops. It hits the brakes until the problem is fixed. And this whole highly regulated, incredibly complex process has one single, absolutely vital goal. And this really gets to the heart of it, the why. Mitosis is the engine of life. It's the reason you can grow from a single cell into, well, you. It's why your body can repair itself. It's the process that ensures continuity, making sure every new cell gets the exact same set of instructions to do its job perfectly. This whole microscopic dance is truly fundamental to our existence. But it really makes you think, with trillions of cells dividing all the time, what happens when that quality control system fails? What happens when just one tiny step in this unbelievably intricate process gets missed? That is when things can go very, very wrong. And that's a story for another explainer.